who work every day towards find a cure for these diseases. The discussion will be open after 3 talks. We have time for the open discussion. Please feel free to ask questions in the chat at any time. When doing so, please don't forget to mention your name and affiliation and to whom the question is addressed. I want also to remember that the webinar will additionally stream it online upon registration. Please, Antonio, introduce our speakers. Antonio? Antonio, can you list me? We have a problem.
Disconnected, I guess.
Antonio? Okay.
He's not connected.
It's not connecting. Okay, we can move on. You think you can, I can?
Sorry.
Okay.
I just touched the wrong button, so sorry, I have to connect again, but I am lucky to have here Angel, and so I am able to talk from his computer. All I want to say is that we are having a very excellent experience with Sapienza University and the Ben-Bedfar Cost Action, teaming up together to organize this webinar series. This is the 5th webinar series that we have, and every time we organize these webinars have been a great success. I think high quality speakers certainly help a lot. And in this case, I want to thank particularly Brigitta Butari, because she has been working very hard to get the best possible speakers for today. So thank you very much, Brigitta. So we will start now with the 3 webinar series, with the 3 seminars. Ana Rojo, Valeria Cordone, and Isabel Lastres, they represent 3 very important aspects of neuroscience, particularly of neurodegenerative diseases related to ALS, Fred syndrome, and tauopathies. So we will start with each of them, starting with Anna, then with Isabel, and then with Valeria, and finally with Isabel. Brigitta, you probably already said, maybe I didn't hear, that they will have between 20 and 30 minutes for the talk, and then we will have a joint discussion at the end of the seminars. So I am very happy to introduce Professor Ana Rojo. Ana Isabel Rojo has been working for many years on neurodegenerative diseases, in particular in Alzheimer's disease, Parkinson's disease, and in the latest years with an independent career on the amyotrophic lateral sclerosis problem, always focused from the point of view of the role of NRF2. So please, Anna, the floor is yours.
Okay, thank you very much for this nice introduction. Can you hear me properly, I guess? So I will share my screen. This one. Okay, can you see my screen?
Yes.
Yes, perfect. So many thanks to the organizing committee to give me the opportunity to stay here today and talk to you about the key role of NRF2 in resolution of neuroinflammation. Although the origin of Parkinson's and Alzheimer's disease is still unknown, the truth is that
Excuse me, we cannot see the presentation.
The presentation.
No? Why?
You have to switch.
This one? No, maybe I will do it again. This one. Okay.
Now we see PowerPoint now.
Okay, now? Okay. So I will start again. We will talk today about the key role of NRF2 in resolution of neuroinflammation. And as probably you will know, the origin of Parkinson's and Alzheimer's disease is still unknown, but there are so many neuropathological features that are shared in common by these diseases and also other neurodegenerative diseases, including the aggregation of toxic proteins, neuronal loss, and the topic of the seminar today is the low-grade and chronic inflammation. There are different types of cells that are involved in this process, but here I want to focus on microglial cells, because these types of cells are considered as the innate immune system within the central nervous system. They develop from your cell as other immune cells within our bodies. They can adopt different type of phenotypes, which are accompanied by different type of morphologies, as I will show you in the next slides. They are involved in a lot of functions related with homeostasis, phagocytosis, surveillance of the brain parenchyma, and very relevant with the synaptic communication and pruning. They have been also associated, of course, with neurodegenerative diseases, including those that we are going to discuss today, which are the Alzheimer's and Parkinson's disease. Very briefly, I want to show you how a microglial cell can get activated. This is a very, how I can say, it's a very plain slide in which I have summarized all the two extremes, all of the microglial activation phenotype. I mean, this is like a type of a spectrum in which we have two poles. One of them is what I call here the M1, or the pro-inflammatory phenotype, and then we have on the other side the M2, or anti-inflammatory phenotype. But just to be for simplicity, I want to show you very briefly how these cells, who is constantly surveilling the brain parenchyma, become activated in a way or in another. It depends on the stimuli that reach their membrane receptors. Okay, if we have a pro-inflammatory stimuli, for example, interferon gamma or lipopolysaccharide, we will have an activated microglia with the M1 phenotype. This type of cell will express a battery of genes related with pro-inflammation. I mean, for example, here you have interleukin-1 beta, interleukin-6, this type of chemokines, some enzymes related with superoxide production, nitric oxide production, okay? And the final stage of this activation phenotype will lead to phagocytosis, okay? On the other hand, if we have other type of activation molecules, such, for example, interleukin-4, 10, or subtype of immunoglobulins, these cells will achieve other type of phenotype, which are more related with an anti-inflammatory phenotype, okay? Because they are going to synthesize, in this case, anti-inflammatory molecules, including interleukin-10, TGF-beta, some pro-inflammatory inhibitors, or other enzymes related with tissue remodeling, because this is the final end of this type of activation, tissue remodeling and bone healing. Anyway, this is the beneficial outcome of the activation of the innate immune system within our brain. But we need to shut down all of these responses, because otherwise they will become detrimental. So these shutdown mechanisms are crucial to restore the microglial homeostasis within our brain. But what is the problem in the neurodegenerative diseases? The problem, part of the problem, of course, is that the trigger never ends, because the damaged neurons are always there. So we have always these M1 phenotype activators molecules impinging this type of phenotype, okay? And the balance between M1 and M2 goes towards a pro-inflammatory response. This is the problem this is the situation in neurodegenerative diseases, because all of this stuff that were beneficial in an acute and controlled way now become neurotoxic. Here you have some examples of production of oxidative stress, with these molecules could be damaging the membrane, the DNA of the neurons. On the other hand, we have a reduction of these neuroprotective functions. So it's crucial to maintain this equilibrium. Due to the fact that reactive oxygen species plays a very relevant role as second messengers within the M1 phenotype acquisition, for example, molecular oxygen could be converted into superoxide anion by the NOx2 system in response to a pro-inflammatory stimuli. Okay? This increase in intracellular ROS will allow the activation of some kinase pathways and finally transcription factors, including NF-κB. Okay, just to amplify, just to lead into the amplification of the inflammation. And at this point, we wondered what about the antioxidant machinery, because if we push the balance here with reactive oxygen species, we wondered what could happen if we reinforce the endogenous antioxidant machinery by the activation of the transcription factor NRF2. As probably you will know, NRF2 is a transcription factor that will transactivate a very strong cytoprotective program, because it is able to activate more than 200 of genes related with different types of processes related with cellular homeostasis, and very relevant here, it is able to modify the specific genes related with immune control and also with redox balance. Here we have some examples, including M-oxygenase 1, which will lead to bilirubin production after different type of reaction, detoxification of semiquinones, or other related with the glutathione system that you know that it is a very relevant molecule to maintain the redox homeostasis. At this point, we wanted to know how NRF2 activation could lead to the modification of N1 and 2 phenotypes within the microglial cells, and we employed an approximation of based on the generation of a dopaminergic damage in mice, based on the inoculation of these animals with a toxin which is MPTP. As probably you well know, in Parkinson's disease in humans, we have a depletion of dopaminergic neurons within the substantia nigra pars compacta. These neurons are part of the circuit that will control the movements of our bodies. And we mimic this dopaminergic neuron loss employing this toxin, which is brain permeable, and could be converted into MPP+, which is the active principle, and can enter specifically in the dopaminergic neuron by the use of the dopamine transporter in these neurons, okay? And then this molecule will inhibit the complex 1 of the electron transport chain within the mitochondria. This will lead to a depletion in the ATP levels and increased levels of reactive oxygen species. So we use this MPTP in mice, wild-type mice, or in mice null for the expression of NRF2. And as I told you here, as you can see here in this picture, as we show in human, but in this case with mice, we can see a reduction in the number of in the dopaminergic neurons when treated with MPTP that was elevated in the case of the NRF2 neuro mice. Here you have the quantification, and the same on the other part of the brain of the animals, because here we have the soma, and here in striatum we have the axons of these neurons. So here you have that the mice treated with MPTP that also have the deficiency on NRF2, we have a depletion, a more profound dopaminergic damage. And it was quite interesting to analyze that in these animals, we observed a misbalance between the M1 and M2 markers of the glial cells, of the microglial cells, because when we analyzed M1 markers, remember the pro-inflammatory markers related with this detrimental activation, in the black bars, they are from the knockout mice, we have increased levels of, for example, interleukin-6, TNF-alpha, or interleukin-1 beta. On the other hand, in the case of M2 markers, those related with anti-inflammatory, wound healing, tissue remodeling, we have a depletion in the knockout mice, and even worse in both cases when we analyze it, the group treated with MPTP. So we have a misbalance when we remove the expression of NRF2, but can we reinforce NRF2 activity to improve these parameters? This is the next question that we want to answer. And in this slide, I want to remind you very briefly that NRF2 is a transcription factor that is constantly synthesized by the ribosome, but it is constantly degraded by the proteasome. Okay, so NRF2 can undergo ubiquitination by this complex, which is led by KEAP1. Okay. And this is what happened in basal conditions. But in response to reactive oxygen species or some electrophiles, this complex becomes inactive, and the newly synthesized NRF2 can enter into the nucleus and heterodimerize with the MAP family members to transactivate the cytoprotective program that I showed you before in the previous slides. So in this context, with this information, we wanted to know what about reinforcing NRF2 activity employing sulforaphane. Sulforaphane is a natural product derived from the broccoli sprouts that could be used in vitro and in vivo, and in this case, we use it in vivo by intraperitoneal injections in mice submitted to this scheme of MPTP intoxication, and with this scheme of analysis of different time points in which we intoxicated the mice during 6 or 3 days, pretreated or not, with sulforaphane. And first of all, I want to show you that we were able to activate NRF2 within the brain because we were able to detect an increase in some of the crucial genes activated by NRF2. Here you have H01, NQ01, or those related with the glutathione system. And in this scenario, we looked for a lot of information related with dopaminergic damage. It was okay. When we treated with NRF2, we were able to detect a reduction in the dopaminergic damage, but on behalf of time, I will skip all of this data. But I want to show you that with that NRF2 activation in brain, we have also a reduction in neuroinflammation. Here you have an immunohistochemistry analysis of the ventral midbrain of the mice. Here you have the part of the substantia nigra, pars compacta, and as you can see here, when we treated these slices with antibodies against IVA1, which is a marker of pro-inflammatory microglial cells, we can see a kind of scar appearing when the animals were treated with MPTP. Here you can see it. Okay, but when we pretreated the mice with the NRF2 activator, we were able to prevent the apparition of this scar. And here we have another analysis, which is complementary, in which we analyze it by coimmunofluorescence analysis, the dopaminergic neurons in green, and in red we have the IBA1 positive microglial cells. And as you can see here, in the MPTP we have an increase in the active microglial cells that were decreased in the case of the sulforaphane treated mice. So we think at this moment that in fact the microglial phenotype acquisition is tightly controlled by the redox homeostasis, because we can pass from the surveillance microglial state to a M1 pro-inflammatory profile, which is characterized by the activation of transcription factor, including NF-κPV, in a first wave. And then we will have NRF2 activation in a second wave, in a later wave, just to transactivate the antioxidant program, maybe, and the other one probably, but trying to organize or to revert this misbalance between ROS and antioxidant machinery production. Okay, in the second part of the talk, I want to move into other neurodegenerative model, because we want to prove that we were able to reinforce NRF2 activity and to modulate neuroinflammation, also in a model in which oxidative stress were not so involved. I mean, we moved into an Alzheimer's disease model, but in this case, we have analyzed the role of NRF2 in neuroinflammation, but in the control of proteostasis. Because as probably you will know, in Alzheimer's disease, we have also neurodegeneration of cholinergic neurons, accompanied by the aggregation of toxic proteins, including amyloid peptide in beta plaques, and also hyperphosphorylation of tau. These two altered process were mimicking a mice model, overexpressing mutant versions of APP, which will lead to the formation of A beta, and overexpression of tau in a wild-type background or in a background without NRF2 expression. We have a lot of data regarding the characterization of these animals. I want to show you only those related with neuroinflammation. But first of all, I want to show you that, in fact, NRF2 deficiency aggravates some of the AD hallmarks, such, for instance, the memory loss. Here we analyze it with this test, which is based on the novel object recognition test. It is a kind of test in which we analyze the memory of the animals, training the mice with two identical objects, and then we remove one of them and put another, and we calculate the time that the animal spent exploring the new object, and we have done a series of calculations to obtain this discrimination index. As you can see here, the animals that do not express NRF2 exhibited a reduced discrimination index. What does it mean is that the animals spend the same time with the old or the new. They didn't remember, okay? So NRF2 deficiency aggravates the memory impairment of these animals. On the other hand, we wanted also to analyze the motor misbalance that these animals develop along of the disease. more related to tau pathology in these cases, and we determined in a motor test some aspect related with coordination, with clasping, with the way in which the animals go for a beam, the back curvature, and we give all of these value between 0 and 3, and we obtain an average, a motor average score. And again, as you can see here, the animals with the deficiency of NRF2 expression exhibited more problems, more motor problems related with this score. Very briefly, I want to show you that in fact these animals, in the brains of these animals, exhibited a reduced capacity to maintain the redox homeostasis because they exhibited reduced levels of glutathione. Here you have the red bars, and on the other hand, some biomolecules such as protein and lipids appeared modified by oxidative stress. Additionally, when we analyzed some pro-inflammatory mediators related with M1 phenotype, what we get was the same as we get in the Parkinsonian model, in which NRF2 deficiency leads to an increase in protein levels of interleukin-6 or one of these two enzymes related with the production of oxidative stress in some extent. When we performed immunochemistry analysis, determining the abundance of activated astrocytes in this case, because we employed antibodies against glial fibrillary acidic protein, GFAP, or in the case of microglial cells, IVA1 in different parts of the hippocampus. As you can see here, in the animals without NRF2 expression, we have more activated astrocytes and more activated microglial cells with an amoeboid phenotype, more related with the M1 pro-inflammatory phenotype that we have quantified. And here we have the quantification of all of this stuff. So again, as in the other model, we were able to unmask the role of NRF2 regulating the neuroinflammatory processes in these mice, and we wondered again if we could endorse a new therapy trying to restore this NRF2 antioxidant machinery. But in this case, we employed dimethyl fumarate, which is a compound that, as probably you will know, it is the only NRF2 activator that has been approved for the treatment of multiple sclerosis and psoriasis. So we treated the animals by oral gabbies, in this case, with vehicle or dimethyl fumarate during 6 weeks, and we performed the motor test that I showed you before in the previous slides, or the novel object recognition test, okay, during this time. First of all, again, we were able to activate NRF2 within the brain because we detected some of the NRF2 transcript by real-time PCR. Here you have the examples, some examples. And then what happened when we go to analyze the neuroinflammation within these mice? Here we have the activated astrocytes or the activated microglial cells within the M1 phenotype. And in different parts of the hippocampus or in other parts of the brain, which is the pontine reticular formation, when we pretreated the animals with dimethyl fumarate, we obtained a reduced number of GFAP positive astrocytes or IVA1 positive microglial cells. We then also perform biochemical analysis, and here you have a reduction in all of this stuff related with M1 pro-inflammatory phenotype. Regarding the acquisition of this motor score, this misbalance in the motor coordination of the animals related with probably with tau pathology, what we have was that in the dimethyl fumarate treated mice, we have a reduction in the acquisition of this motor score. So we have an improvement of these motor problems with dimethyl fumarates. And something similar happened with the discrimination index of the novel object recognition test. We have a slight difference, but during at least during the week number 2, it was significant, and we have a reduction. In this case it's the red. We have an increase in the discrimination index, showing a better memory performance of the NOR test of these mice. So we think with all of this data and all of data that has been published during these years in the bibliography, that in fact NRF2 has a very relevant role in the resolution of inflammation, because it will be able to transform, to maintain this equilibrium, this balance between the neuroprotection exerted by a physiological activation of the immune response within our brain to neurotoxicity induced by the pro-inflammatory microglial cells, aggravating the neurodegeneration. And then there has been described a lot of different types of molecular mechanisms that could be involved in this resolution of inflammation of NRF2. Some of them has been related directly and other indirectly. Directly, we have some examples in which when NRF2 is bound to the antioxidant response elements or of crucial anti-inflammatory cytokines, it could develop this type of M2 phenotype. For example, this receptor is related with phagocytosis or Marco or this interleukin, okay? Or maybe it could lead to the inhibition of the interleukin-1 beta and interleukin-6 production by the inhibition of the binding of RNA-POL2, as Masayuki Yamamoto group has demonstrated. And on the other hand, we have the indirect direction, okay, because in the way that NRF2 will reduce the RUS production, all of this, when we inhibited this generation of oxidative, reactive oxygen species, will lead to a reduction in all of these pathways activated by them as second messengers. And on the other hand, there is also some kind of inhibition because HO1, which is a classical target of NRF2, could inhibit the expression of these molecules which are involved in migration and infiltration of other immune cells within the brain. So on, and all of this data really support that NRF2 has a very relevant role in the resolution, in the inflammation resolution. Finally, I want to thank you, all of you, for your attention, again, for inviting me to give this talk, and all of our team. Thank you very much. I will be happy to answer your questions.
Thank you very much, Anna.
Okay, let me come back.
Yes, I'm back. So thank you very much. We will leave the questions for the end of the session. And now we will go to the next speaker, which is Valeria Cordone. Valeria is a postdoc in the laboratory of Giuseppe Valacchi, and she has been working for the past years on something that she will explain called oxy-inflammation. Valeria, thank you very much for being here. The floor is yours.
Thank you very much for asking me to participate to this Redox webinar series. And thank you for the presentation. I'm going to share the screen. Okay, can you see the screen?
Yes, we can, but can you put it in full screen?
Okay, perfect. Okay. Okay, so
Now better.
okay, perfect. So today I'm going to talk to you about the NRF2 involvement in neurodevelopmental pathologies, Rett syndrome, and CDKL5 deficiency disorder. Okay. Before to start, I will briefly introduce the history of these disorders, because maybe not all people knows these rare pathologies. Rett syndrome was first described in 1966 by the pediatrician Dr. Andreas Rett, who reported in German the findings of 22 patients. But just in 1983, Dr. Ackberg reported other findings in English, so the syndrome could be recognized further. The previous version of the Diagnostic and Statistical Manual of Mental Disorders, Rett syndrome was classified as one of the autism spectrum disorder. However, in 2013, with the new DSM-5, Rett syndrome has been no longer included among the ASD, so it became an independent pathology. However, despite this, the link with autism still remains. Indeed, many children are diagnosed as autistic before the MECP2 mutations, since Rett syndrome as even a genetic component are identified, and then the diagnosis is revised to Rett. About the CDKL5 deficiency disorder, even in this case, it became an independent disorder in 2013 because previously was part of the atypical form of Rett syndrome, in particular the early onset seizure variant of Rett syndrome. Okay, as already mentioned, the Rett syndrome as a genetic component. Indeed, it is caused in the 95% of the cases by mutations at the X-linked gene methyl-CpG binding protein 2. This protein is an ubiquitous protein present in two different isoforms, the E1 and the E2. It contains several functional domains, the MBD, methylated, a functional domain for methylated CpG dinucleotides, and the TRD, which contains a domain for the transcriptional repression, and then the C-term domain, which helps the protein to bind DNA. To date, over 800 different mutations are known to cause different types of Rett syndrome. But which are the functions of MECP2? MECP2 is an epigenetic reader involved in several different cellular processes, and in particular in transcriptional repression, but even in transcriptional activation, then even in chromatin loop formation, in chromatin compaction, and in miRNA processing. But what about CDKL5? Similarly to MECP2, CDKL5 is an X-linked gene, and it encodes for a serine threonine kinase capable of phosphorylate itself and other target proteins, including MECP2. CDKL5 is required for dendrite growth and dendritic arborization. And to date, over 200 variants of likely pathogenic CDKL5 mutations have been identified. Okay, so what can make these pathologies even more complex? The issues of the X-linked disorder, so which is the X chromosome inactivation. X chromosome inactivation, as we know, is a strategy to equalize the X-linked gene dosage between XX females and XY males, and involves the transcriptional silencing of the genes of one X chromosome in females. So the X chromosome inactivation is often random, and so either the maternally or paternally inherited X chromosome can be silenced. So Rett syndrome patients as well as CDD patients present a mosaic pattern of cells due to X chromosome inactivation, which means that part of cells express the wild type allele of MECP2 or CDKL5, and another part of cells will express the mutant forms. Okay, now let's focus our attention on Rett syndrome. Rett syndrome is a devastating neurodevelopmental disorder which affects the female gender with a prevalence rate of 1 per 10,000 live births. Clinically, after a 6 to 18 months of apparently normal development, the disease leads to an early neurological regression. Clinically, the pathology is classified in 4 different stages. Stage 1 is defined stagnation. Stage 2 is defined the rapid regression, where usually patients undergoes loss of acquired motor skills and communications. There are the appearance of seizures, microcephaly, and breathing irregularities. Then there is the stage 3, which is a pseudostationary period, and then the final stage with the complete motor deterioration. However, in addition to all the non-neurological manifestations, Rett syndrome patients even show multi-system comorbidities. Indeed, to date, it is defined a broad spectrum pathology, not only a neurological pathology. Some common comorbidities include breathing disorders, sleep disturbances, cardiac abnormalities, gastrointestinal problems, osteopenia, and even recurrent infections. Furthermore, in the last decades, several papers reported the compromised metabolic processes. First of all, Rett syndrome show an persistent unbalanced redox homeostasis, which is characterized by reduced antioxidant defenses, both enzymatic and non-enzymatic, in parallel with the increased redox imbalance, characterized by an augmented mitochondrial ROS, hydrogen peroxide, as well as 4-hydroxynonenal protein adducts. Then, coherently with the situation of oxidative stress, Rett syndrome patients show even mitochondrial dysfunctions, consisting of an abnormal mitochondrial morphology, an impaired mitochondrial quality control, and mitophagic processes. At the end, Rett syndrome patients even present a chronic subclinical inflammatory state, as evidenced by all these papers. This pro-inflammatory state is characterized by the regulation of acute phase response proteins, a disturbed cytokine profile, an atypical morphology of lymphomonocytes, and an aberrant NLRP3 inflammasome activation. Importantly, and even more interestingly, maybe, it has been shown that the oxidative stress and inflammatory markers are increased since the earliest stage of the disease, here, as you can see here, and even in a pre-symptomatic phase of Rett mice. So together, these compromised metabolic processes characterize a condition known as oxinflammation, which is able to act in a vicious circle and induce damages at several organs and tissues. So say that, in light of the inflammatory state, what happens at the microglial level? It seems that even microglia of Rett syndrome patients are involved in the oxy-inflammatory status. Indeed, it has been shown that microglia from MECP2 null mice is involved in the release of abnormal high levels of glutamate with neurotoxic effects on hippocampal neurons. Then even a transcriptomic study on microglia from Rett mice at prephenotypic stages revealed a dysregulated expression of genes associated with innate immunity and cellular stress responses, suggesting that this alteration could be the prelude to the later onset of neurological symptoms. Now I'm going to focus the attention on this interesting paper published in 2021, where the authors used an evolutionary genomic approach to perform an unbiased MACP2 gene network using over 1,000 different eukaryotic genomes to prioritize proteins coevolutionarily ligated to MACP2. Among all the proteins, they focus their attention on KEAP1 and IRAK1. As the Professor Hoho said, KEAP1 is the cytosolic repressor of NRF2, while IRAK1 is a protein downstream of toll-like receptor and upstream of the NF-κB activation. So then they used an inhibitor of IRAK1, which is the pacritinib, and the dimethyl fumarate, which allows the NRF2 dissociation from KEAP1, thus is considered an indirect NRF2 activator. And as you can see from the graph, both these molecules were able to abrogate only partially the effects of MACP2 silencing on human immortalized microglial cells. And then the treatment of these silenced cells with the dimethyl fumarate exerted a significant increase in the phagocytotic capacity of microglia. And at the end, the DMF was even able to downregulate the increased NF-κB activation in MACP2 knockdown microglial cells. So the authors proposed that the lack of MACP2 can bring to an increase of IRAK1 and KEAP1, which together can act indirectly or directly to the NF-κB activation in this syndrome. Actually, this is not the only paper reporting the involvement of NF-κB in this pathology. Indeed, we recently published two papers showing an increased nuclear localization of P65 in both primary fibroblasts and lymphomonocytes of isolated from Rett syndrome patients. In addition, the knockdown of MACP2 has been reported to lead to increased NF-κB even in neuronal and myeloid cells. And this deregulated activity has been associated with neuroinflammation and decreased dendritic arborization in a mouse model of Rett. So, as already said by Professor Rojo, the NF-κB is strongly interconnected with NRF2, and this interplay can occur at a different level. First of all, at the transcriptional level, because there is a KB responsive element in the promoter region of NRF2. Then it can occur at the about the competition for CBP P300, and at the end, in a response to ROS overexpression. So in light of this interplay between NF-κB and NRF2, and in light of the oxyinflammation, the aim of one of my current projects is to investigate the crosstalk between these transcription factors in fibroblasts derived from Rett patients and control subjects, and stimulated with lipopolysaccharide or hydrogen peroxide as a pro-inflammatory and a pro-oxidant stressor. Unfortunately, I cannot show you the results because they are preliminary and they are not published yet. But however, I can briefly summarize the key findings. So we saw an increase of nuclear NF-κB P65, but even an increase of NRF2 levels in Rett cells already in basal condition. while LPS treatment was able to induce an increased translocation of both these transcription factor only in control fibroblasts, but not in red cells. While hydrogen peroxide treatment induced an enhanced translocation of NRF2 into the nuclear compartment of control cells. Conversely, red fibroblasts were not able to elicit a protective NRF2-related response against the hydrogen peroxide, as confirmed even by the unchanged levels of HO1 mRNA, hemoxygenase 1. While the red fibroblasts seem to promote the nuclear translocation of P65 as a first reaction to hydrogen peroxide. Then, to the best of my knowledge, there are two other papers investigating the role of NRF2 in Rett syndrome. These papers are published by the same group, and they used a 1-year-old fully symptomatic Rett mice treated with metformin for 10 days. They saw that Rett mice exhibited an increased hydroxynonenal adduct, and metformin was able to reduce this oxidative stress marker. Then, about NRF2, the authors saw an increased level of this transcription factor in Rett mouse hippocampus. And then they even evaluated the phosphorylation of NRF2 as an index of its activation status, and it was increased due to metformin treatment. However, the improvement in the oxidative stress status due to metformin was not mirrored by the beneficial effects on the behavioral phenotype. So the authors decided to change the treatment and performed a longer administration of metformin for 16 weeks. And first of all, they confirmed that an upregulation of phospho-NRF2 in Rett mice upon metformin. However, they could notice only a selective normalization of cognitive flexibility in Rett female mice, and not an amelioration of the other parameter of general health and motor dysfunction. So they hypothesized that the effect of NRF2 and metformin may be more good at early phases of Rett respect to fully symptomatic later stages of Rett syndrome. Okay, now let's switch our attention on the other pathology, the CDKL5 deficiency disorder. CDKL5 deficiency disorder, CDD, is even more rare than Rett syndrome. Indeed, its incidence is 1 per 40,000 to 60,000 newborns. And it is characterized by seizure onset before 3 months of ages, and then by the compromised language and hand function skills. Both Rett and CDD are characterized by several common manifestations, and other common denominators at molecular level are the perturbed cholesterol homeostasis, as you can see here, and at the total cholesterol, LDL, and HDL levels. Then an increased cellular and systemic oxidative stress is characterized even by mitochondrial dysfunction abnormalities, and an aberrant microglial activation and a subclinical attenuated inflammation in the systemic compartment. So in this paper published by the group of Professor Valacchi, the authors focused their attention on a specific receptor for HDL, the SRB1, and they found that this receptor was significantly reduced. In parallel, they even confirmed the oxidative stress and nitrosative stress in cells derived from CDKL5 deficiency disorder patients. So, and then CDKL5 fibroblasts even showed an increase for hydroxynonenal and nitrotyrosine SRB1 adducts that lead to the ubiquitination of this receptor, and so maybe to its probable degradation. So this loss of SRB1 can be associated with increased lipoprotein found in the serum of CDKL5 patients. So in light of this high oxidative stress in fibroblasts derived from these patients, the authors focused their attention on NRF2. And as you can see from here from the graph, CDD cells had lower levels of NRF2 and even in the nuclear compartment. And in addition, these cells were unable to induce a further translocation of NRF2 in response to hydrogen peroxide or H&E hydroxynonenal stimulations. And this was confirmed even by the PCR of GCLC gene. Okay, this is the first report showing in CDKL5 patients an increase in both oxidative and nitrosative stress marker and an NRF2 aberrant response. Together, they are all factors that can contribute to cell damages. What can we say at the end? There are several findings revealing a common perturbed pathways related to redox homeostasis, mitochondrial dysfunction, and inflammation between RET and CDT. The NRF2 role in these in the neuroinflammation and pathogenesis of these neurodevelopmental disorders is still very poorly investigated. However, some evidence seems to indicate that this transcription factor, NRF2, may represent a good target for the development of new possible therapeutic strategies for these devastating disorders still without a cure. Thank you for your attention, and thank you so much.
Thank you very much, Valeria. I just wanted to say, I forgot to say that Dr. Valeria Cordone and Professor Giuseppe Valacchi are located at the University of Ferrara in Italy.
Yes.
Now we are going to move to the next speaker. This is Professor Isabel Lastres-Becker. She is a professor at the Department of Biochemistry at the Autonomous University of Madrid and also works at the Institute of Biomedical Research. Isabel has been working for many years on, actually, I think her entire career on issues related to neurodegeneration. And for the past maybe 15 years or so, you will say, you have been focused on NRF2, which is great, because it gives a big plus to the field. Isabel, whenever you want. Thank you.
Thank you very much, Antonio. And do you see my presentation and the pointer?
Yes.
Thank you. Okay. Thank you also to the organizers to giving me the opportunity to share with you our results in relation to the chemokine receptor CX3, CR2, R1, and NRF2 in relation to tauopathies and also inflammation. And in relation to tauopathies, there is a broad spectrum of diseases that can be implicated in these pathologies, but we are going to focus on frontotemporal dementia, which is also a tauopathy, as we will see. And maybe you have heard the last news that Bruce Willis nowadays is suffering this devastating disease. And frontotemporal dementia there is characterized by the degeneration of the frontal and temporal lobe, and also followed by hippocampus atrophy. And it can be characterized at different point of views into different kind of diseases as behavioral variant and also as a primary progressive aphasia. And in both cases, they are follow up by a hippocampal atrophy. And the most important point of this disease is that there is a very early kind of dementia, because it's the most common cause of dementia in people younger than 65 years, in opposite to Alzheimer's disease, which dementia just started when the people get older than these years. But in general, all these neurodegenerative disorders, the most common and most important thing is that nowadays they are not cured, and that's why we're focusing in this pathology. And in relation to neuroinflammation, there is also an implication of two proteins like tau protein, which is associated with mutation in these genes. For example, tau mutation P300L and P300S, which is associated with frontotemporal dementia. In the other way, we can focus on TDP43 positive inclusions in these patients, and there are also mutation in these genes associated with this disease. In our lab, we are working with both models, but the results that I'm going to show you are based on the tau disease-based model. And in this case, in both cases, TDP and tau dependent frontotemporal dementia, and it has been observed that there is an alteration in glial phenotypic landscape associated with this chronic inflammation, where there is an alteration in the phenotypes of the microglia, and the phenotype of the microglia, as we have heard in the first talk that Ana Rojo just gave us, you have different subtypes, and also there are association of different kind of glial phenotypes associated with different mutations and regions in the brain associated with glial and microglial activation. And also there is an activation of astrocytes related to the neurodegenerative process, which also exacerbate the neuronal death. Moreover, there are also other relations about inflammation and frontotemporal dementia. It has been observed that there is an upregulation of several inflammatory markers, also a decrease in levels of anti-inflammatory markers. And in some cases, it has been demonstrated there is some kind of autoimmunity in this disease, where different kind of genetic mutation associated with frontotemporal dementia are linked to autoimmune conditions and also to different kind of genetic mutations and the presence of several kind of antibodies in this kind of patients. Moreover, there is an alteration in the blood-brain barrier, which just exacerbate also the penetration of different pro-inflammatory products within the brain. And this dysregulation of inflammation just are associated with the development and also the impairment of the neurodegenerative process. Moreover, there are several kinds of genes that are associated with the genetic link of the disease that are implicated in inflammation processes. For example, TREM2, that maybe you have heard, that is a marker of microglial cells. In general, so there is a kind of international crosstalk between inflammation and frontotemporal dementia. That's why in our lab, we focus on one kind of chemokine, which is called frataxin, which has also the name as CX3CL1. I'm always going to talk about frataxin. And before our results, it has been shown that when a neuron is dying, frataxin is increased the expression, and depending of the form of frataxin, there are two kinds of functions. When it's bound to the membrane, the fractalkine has a function as an adhesion molecules. However, this protein can be cleaved by ADAM10 and ADAM17 and became soluble, and then the function changed and has a chemioattractive activity. In this case, when fractalkine is soluble, can bind to its receptor, which within the central nervous system, the receptor is located specifically in microglial cells. Then this soluble fractalkine binding to its receptor activates phagocytosis and also is able to modulate microglial response. All this system belongs to what is called find me, eat me, and digest me system. In this case, fractalkine is in the first step where microglia has to find the neuron that is dying in the function of chemioattraction. And it has been postulated that this activation of microglia has belong other activities also as an inflammatory function. Therefore, in our lab, we were interested to see the relation between this system and the tauopathies, in this case, with the tau mutation P301L, which, as I mentioned, is a kind of genetic form of frontotemporal dementia. And what we have observed is just overexpressing tau in the hippocampal cell line, HT22, expressed by lentivirus. We observed that there was a change in the morphology also of the neurons, and this tau overexpression neurons just increased the mRNA expression of fractalkine and also the soluble form of this chemokine. Related to microglia, we wanted to see whether this fractalkine was able to activate these anti-inflammatory processes, just modulating the transcription factor NRF2. And in this case, as you have heard in the previous talks, NRF2 can regulate many genes, also associated with inflammation. And in this case, we just observed that giving recombinant fractalkine to the microglia, NRF2, the mRNA levels of NRF2 were not changed. But when we looked at the protein levels, we just see clearly that there was a translocation of NRF2 to the nucleus, and this translocation of NRF2 to the nucleus induces the expression of NRF2 dependent genes as hemocyclinase 1, glutathione peroxidase, GCLC, and GCLM at mRNA levels and also at protein levels, indicating that fractalkine was able to activate NRF2 in microglial cells. To determine if this was a direct effect of the fractalkine on NRF2 signaling pathway, we just analyzed the same system, but in this case, within microglial cells, primary microglial cells, obtained from wild type or NRF2 deficient animals. As you can see, in NRF2 deficient animals, there was always a decrease, as expected, in NRF2 dependent genes, and the treatment with the recombinant fractalkine just induces only in NRF2 wild type microglia, but not in the NRF2 deficient microglia, indicating that this system was directly associated with the activation of NRF2. Moreover, you could think maybe these microglial cells could have less CX3CR1 receptor, and indeed we measured the mRNA levels of the receptor of fractalkine, and there was no change, indicated that this effect was really due to activation of NRF2. Moreover, after the Find Me system related to fractalkine, there was the Eat Me analysis pathway in which there are several scavenger receptors implicated. Belonging to these scavenger receptors, they are the family of the TAM receptors, which are composed by 3 different kinds of tyrosine kinase receptor, TYRO3, AXL, and MER. And all these receptors function in the same levels. When you have a neuron that is dying, you have a flip of phosphatidylserine, And this phosphatidylserine can be recognized by several proteins, as for example, Gas6 and protein S. And these proteins function as a link to these TAM receptors, which can be found in the microglial cells within the brain. And in this case, what we observed in relation with this TAM receptor was that in the microglial cells of NRF2 deficient mice, all these three receptors were downregulated, indicated that there was also not only at the find me signaling pathway an alteration in the NRF2 deficient microglia, but also in the next step, the eat me signal system. Moreover, in relation to phagocytosis, we observe that the deficiency of NRF2 has also an impairment in the phagocytosis of the microglia, indicated that NRF2 deficient microglia has an overall impairment in the function. That was at the cellular models, but we wanted to see what's happening in a tau, an frontotemporal dementia tau-based model in mouse. And in this case, we collaborated with the laboratory of Fred van Leeuwen. And in this case, we just injected within the brain an adeno-associated virus that will overexpress tau with the point mutation P3100L under the control of synapsins, which indicates that tau will only express in neurons. Moreover, this model is very useful because within the same animal, you have the contralateral side, which serves as a control, and then you have the ipsilateral side where you have the overexpression of tau. And moreover, you can express this virus in every kind of animal what you want. And in this case, we just injected the virus in NRF2 wild-type mice and NRF2 deficient mice. In both cases, the overexpression of tau was very similar, but what we have seen is that this tau overexpression induced microgliosis in the ipsilateral size in the wild-type animals, and this inflammation related to microgliosis and astrogliosis was exacerbated in NRF2 deficient mice, as we have heard in the previous talks, that NRF2 deficient always exacerbate neuroinflammation within the brain. Moreover, we wanted to see in which kind of cells NRF2 was induced, and what we saw is that in the tau-positive neurons, there was not NRF2 activation, and for that we use hemosigenase 1 as a gene, as an NRF2-dependent gene of NRF2. And when we looked at microglia with EVA1 marker, we saw that there was an overexpression of hemosigenase 1, indicated that in microglia there was an increase of NRF2 activation, and in the same case in astrocytes, where we also see induce of hemosigenase expression. On the opposite, NRF2 deficient mice did not have this hemosigenase induction, which was as expected because there was not an induction of NRF2. Furthermore, we analyzed what's happening in vivo with the chemokine fractalkine, and what we observed was that in tau-positive neurons, there was also an induction of the protein expression of fractalkine, but in this case, we didn't observe changes in the expression neither of fractalkine or the receptor between both phenotypes, which can be also expected because it's upstream the signaling pathway of NRF2. But what happens if we just get rid of the receptor for fractalkine? This means what happens if we have a knockout for CX3CR1 receptor? And in this case, these animals have been developing just inserting the green fluorescent protein within the gene. This means that disruption of the receptor by GFP makes that the cells that doesn't express the CX3 receptor are fluorescent for the green protein. And in this case, we just developed exactly the same model, just injected the adeno-associated virus, just overexpressing tau P300L in CX3R1 positive animals and the deficient animals for this receptor. And in this case, what we observed was that in the wild type animals, we have a tau overexpression induced and microgliosis as expected. And what was very shocking for us, it was that in wild type animals, these microglia express hemoxygenase 1, as we have seen previously. And here you can see the clear microglia, positive microglia overexpressing hemoxygenase 1. But in the other way, when we have the knockout mice for the receptor, this microglia was not able to express hemoxygenase 1. These red cells were astrocytes. This means that what we have seen is that for one side is that the deficiency of this receptor increases neuroinflammation in the same way as NRF2 deficient animals, and the other way that normally hemoxygenase positive cells were exacerbated in the macroglia when we have overexpression of tau, but in the animals that were deficient in CX3R1 receptor, there was no induction of hemoxygenase 1, indicating that there was not induction of NRF2. And finally, of this part of the work, we wanted to see what's happening in a tau in humans in a tauopathy disease. Unfortunately, it's not possible to get so easily samples of frontotemporal dementia, and in this way, we obtain human brain samples of Alzheimer's disease, which is a secondary tauopathy, and also from PSEP, which is another tauopathy. And in both cases, we have seen that there was, in both diseases, there is an induction of the frataxin expression at the protein levels, and there is also an induction in NRF2 signaling. Moreover, when we looked in this patient, when there is a tau positive neuron, these tau positive neurons express frataxin, and these frataxin positive neurons are surrounded by activated microglia. Indeed, what we have found until now is that tau overexpression neurons express and release this chemokine frataxin. This frataxin is able to activate NRF2 pathway, and as similar as what's happening in NRF2 deficient mice, the deficient mice for this receptor behave in the same way. They have an exacerbation of neuroinflammation. And moreover, what is more interesting is that CX3CR1 deficient microglia do not express hemoxygenase 1 as are reported of NRF2 signaling. That led us to think about what would happen if we just could activate NRF2 in these animals, for example, just using sulforaphane as a well-known inducer of NRF2. That's why we wanted to see what happens. Could we restore hemoxygenase 1 levels in these knockout animals? And also we wanted to address the question, which is the implication of this receptor regarding NRF2 signaling, and furthermore, which is the implication of this receptor in the phagocytosis process. For that, we have previously seen also in this mouse model that another activator of NRF2, dimethyl fumarate, was able to exert neuroprotective effects, also have antioxidants modulation, and has anti-inflammatory activity. Therefore, we just treated our mouse model, in this case, in a wild type and in the knockout of the receptor animals, with sulforaphane for 3 weeks intraperitoneal. And in this case, what we observe is that the animals that overexpress tau have an induction of astrogliosis, as observed with the GFAP marker. And furthermore, these animals treated with sulforaphane, we clearly see an induction in the contralateral side of hemoxygenase 1, which was expected. We are inducing NRF2 signaling. And very interesting was that this microgliosis induced by tau was reversed, as we can also see here in the quantification. What's happening in the knockout for the receptor of frattarkin? In this case, we have also a very nice astrogliosis induced by tau. And when we treated the animals with sulforaphane, we could reverse this. This means that at least for the astrogliosis, sulforaphane treatment was able to restore the astrogliosis to control levels. And what's happening to microglia? Because as you remember, the microglia of these kind of animals, the knockout animals, didn't express hemosigenase 1. In the wild-type animals, the overexpression just induced the microgliosis, and the treatment with sulforaphane was able to restore it more or less to control levels. However, in the knockout animals for this receptor, the treatment with sulforaphane was not able to modulate inflammation. We were just shocked because we didn't expect that at least we have some kind of positive results. But you will see why we didn't saw positive results regulating microgliosis in these animals. In relation to neurodegeneration, we use a plasticity marker, which is called calvindin. Calvindin is a calcium-dependent protein that is downregulated when there is an overexpression of calcium and there is a cell death. That means when we have a tau overexpression, we have a very decrease of calvindin expression in these animals, and the treatment with sulforaphane was able to restore the expression of calvindin. So here you have calvindin negative tau positive neurons, and in the case of sulforaphane treatment, you have calvindin and tau positive neurons. When we have the receptor knockout animals, calvindin disappears, as what's happened is also in the wild-type animals, but the treatment with sulforaphane could only restore a little bit, not so much, indicating that the neuroprotective effect that we have seen in the with the treatment of an NRF2 inducer as sulforaphane was mainly related to the microglial activity. Looking for what's happening in the microglia of these animals, the receptor knockout animals, what we saw is the knockout animals have a decrease in the expression of NRF2 signaling at the mRNA levels of NRF2 and other enzymes depending on NRF2 as MQ1, GCLC, and GCLM. And the treatment with sulforaphane, which in wild-type microglia was able to induce NRF2, was not able to induce neither all the NRF2 dependent enzymes. But this was specific for microglia. In astrocytes that came from the same animals, we see that this astrocyte doesn't express the receptor. There was no problem because sulforaphane was able to induce NRF2. Furthermore, we wanted to see what's happening in these animals also with the TAM receptors, and very similar results that we obtained with these animals, but is what we saw in the NRF2 knockout mice. This means we see a decrease of AXL, MER, or TRO3 expression and the knockout of the receptor, similar at what we have shown in the NRF2, the deficient microglia. Furthermore, when we treated the microglia with sulforaphane, only AXL was induced by the treatment with sulforaphane. This led us to investigate whether this receptor could be an NRF2-dependent gene. And just in collaboration from Dr. Natalia Jimenez, we performed a bioinformatic analysis. And in this case, we saw that AXL has added sequence in the promoter, and in collaboration to Professor Heike Allgeier from München, we obtained the promoter region of AXL and just the transfection of NRF2 plasmid that lacks the KEAP1 binding sequence show a dose-dependent induction of this receptor, very similar to what we saw with the treatment with sulforaphane, the NRF2 inducer. And on the other hand, fractalkine activation of this receptor was also observed after 24 hours of treatment, indicating that AXL was an NRF2 dependent gene. Besides TAM receptors, also microglia is implicated in phagocytosis, as we have seen previously. And in this case, what we saw is that the microglia that lacking of the receptor behaves exactly the same as NRF2 deficient microglia. If there is an impairment in the phagocytosis, and fractalkine, which induces the phagocytosis in normal microglia, was impaired in these animals. In the other way, we are also analyzed migration of the microglia, and as you can see, in all cases, microglia deficient in this receptor has an impairment in migration, and neither sulforaphane, fractalkine, or combination of both systems was able to induce the migration of these cells. So just taking together, what we have seen is that the activation of the system of fractalkine and its receptor induced the modulation of the NRF2 system, just activating anti-inflammatory pathways, TAM receptors, and phagocytosis, which modulates the inflammation related to a tauopathy model of frontotemporal dementia. However, when we have no receptor available, the deficiency of these receptors impairs the expression of the NRF2 system and also the expression of anti-inflammatory pathways, TAM receptor, and phagocytosis as NRF2 deficient animals, and this leads to an increase in pro-inflammatory markers and pro-inflammation in the tauopathy mouse models. Altogether, what we know nowadays is that this system behaves in two waves. In the first wave, the activation of the system induces NF-kappa B, and this induction of NF-kappa B in a second wave modulates NRF2. And when we don't have the receptor, there is a decrease of P65 and also a decrease in NRF2, leading to a decrease of TAM receptor and phagocytosis. And what is this and the repercussion, which is the meaning for the pathologies, for example, as frontotemporal dementia or Alzheimer's disease, which also are secondary tauopathies? Indeed, that there are not mutations in NRF2 or CEST of the fractalkine receptor that leads to the pathology, but there are several papers that report that there are haplotypes in NRF2 that influence the progression of the disease. This means when you have less NRF2, you have an increase in the progression, an exacerbated progression of Alzheimer's disease. And also, for example, there are several SNPs in NRF2 that are associated with vascular stiffness with aging. In relation to frontotemporal dementia, there are nowadays no studies relating NRF2 and this disease, but it has been observed that mutations in P62, which is a variant gene associated with this disease, impacts on NRF2 pathway, and is also associated with the development of the disease. In relation to the receptor of fractalkine, it has been reported two different kinds of polymorphisms. One of them is associated with the low availability of the receptor in the cell surface, and the other one is associated with a reduction of the affinity of the receptor for his ligand. And in both cases, it has been associated this kind of mutation, this kind of polymorphism, also within the progression of several pathologies, for example, Alzheimer's disease and also ALS. All this data just indicate that pharmacological modulation of NRF2 could be very beneficial for several kinds of pathologies from frontotemporal dementia or Alzheimer's disease, although depending on there are several polymorphisms that they will be not beneficial. Therefore, we have to think more to personalized treatments on Alzheimer's disease and also frontotemporal dementia that previous the treatment with this kind of pharmacological activators of NRF2 has been first seen if there is a kind of polymorphism that can be associated with the disease. And finally, I would like to thank Antonio Cuadrado because this study just started when I was finishing my stay in his lab. These are my 3 windows in the Instituto de Investigaciones Biomédicas, and these are my current students in the lab. And I would also like to thank our collaborator, Ángel García Yagüe, Sebastián Puiglia, and other collaborators. And thank you very much for your attention, and I will be glad to attend your questions. Thank you.
Thank you very much, Isabel, for this nice talk. A lot of data, but also very, very interesting. So before everybody, for the questions, many people wants to go away. So I just wanted to make a couple of comments on Valeria's talk. I think it is important for everybody. One of them is that electrophilic molecules don't seem to release NRF2 from KEAP1. Apparently, this is a sort of old-fashioned concept now. Now we believe that these molecules disrupt the KEAP1 NRF2 interaction, but don't release NRF2. What happens is that the interaction with the rest of the E3 ligase complex is not proper, is not well organized, and then let's say that KEAP1 remains clogged with NRF2. And it is the newly synthesized NRF2, the one that apparently is moving to the nucleus. It is not important from the point of view of the end result, but from the mechanistic point of view, it may be interesting to know this. And the other thing is I would like to put a word of caution about the use of the phospho NRF2 antibody that recognizes the serine 40, serine 40 phosphorylated NRF2. The problem is that if these people, because it was not your work, it was actually you were mentioning other people.
Yes.
So if you see an increase in global NRF2 levels, and you also see an increase in global phospho NRF2 levels by just Western blot, it is very difficult to tell that indeed you have an increase in the phosphorylation of serine 40. There are other methods that they could use, like maybe immunoprecipitation, maybe some sort of knockout, because there is a feeling there is a general feeling that indeed serine 40 is not clearly relevant for the NRF2 dependent degradation, the KEAP1 dependent degradation of NRF2. This is just a couple of words before everybody runs away. And now we have some very interesting. Valeria, would you like to comment on this?
Yes, thank you for your comments and for your precious information. Yes, I agree with you. Unfortunately, the papers that I showed are not my papers because I'm studying now the crosstalk among between NRF2 and NFKB, and so thank you for your suggestion.
Sure, no problem. So we have some very interesting questions in the chat, and we still have a bit of time to go over them. So I will go just by order according to the first question that were raised in the chat. So for Dr. Ana Rojo, they say, excellent talk. NRF2 effects would likely promote glutathione of inflammasome proteins such as NLRP3, ISC, or NEC7, which would be inhibitory. Have you investigated glutathione levels or protein glutathione?
Okay, thank you very much for this nice question. We didn't analyze specifically the glutathione of these specific proteins, and it would be very interesting, but at this time we didn't do it. What we did, at least in the Alzheimer's disease model, was to analyze the glutathione levels in the brain of these animals. And as I told you before, the Alzheimer's disease model that didn't express NRF2 exhibited reduced glutathione levels within their brains, and on the other hand, exhibited higher levels of biomolecules that could be damaged by oxidative stress, such as carboxylated protein, and peroxidized lipids.
Okay, so thank you. I think as far as I know, glutathionylation of the inflammasome is something still very open to research. And I think that only the ASC protein was shown to be glutathionylated, and then there is a decrease in the inflammasome activity. But I think there are just a couple of papers on this, so who knows? We need to see what's going on in the future.
So, Antonio,
Okay.
I guess that we have another question previous to this one, which is related with Noxis produces superoxide on the outside of the cell. How does it enter into the cell?
I don't see it, so go ahead.
Christopher Ling, I guess, yeah. No, superoxide anion, it's a very short-living species, so probably it should be converted into peroxide, hydrogen peroxide by the superoxide dismutase 3, which is coupled within the membrane, and then it can enter into the cell by aquaporins or something like that. It is not so clear, but the combination of these two oxygen species will be crucial to maintain the oxidative burst of the microglia, in this case of monocytes in the periphery.
Okay, now the next question, this time it is again for Anna. Anna, did you observe any behavioral change like catatonia, walking, etc. in MPTP administered control and NRF2 knockout animals?
Yes, we did observe all of this type of, not catatonia because they were alive and there were the intoxication with MPTP was done in a chronic way in which we have sustained survival. But when we analyzed the motor behavior of these animals employing open field tests, we detected a reduction in the number of crossing lines within the open field test, and also in the times that the animals get rise, the rear, okay, the number of the rears that the animals get with MPTP. But we didn't include this data within our study. It was we did this type of analysis just to check that the MPTP intoxication was right.
So probably Gulay Etzer, who is the person who made the question, knows that the MPTP model is a bilateral model. So in this case, asymmetric motor deficits cannot be easily seen. We have been working with other models, particularly adeno-associated induced expression of alpha-synuclein. And then we see a contralateral effect on motor behavior, particularly in the you hang the animals by the tail and you see the torsion that is contralateral. And this can be done. It is a very sensitive test, but it is in the case of an hemi-Parkinsonian model, not in the MPTP model. Okay, so what else? What else we have here? This is Brigitta asking to Valeria. Valeria, just a curiosity, can different mutations at the MECP2 gene determine a different phenotype of the pathology?
Yes, thank you for the question. Yes, due to the time of a webinar, I could not explain everything about the Rett syndrome. However, yes, MECP2 mutations can be classified as nonsense, missense, and large deletions, depending on where the mutations are in the domain that binds the DNA or in other important domain. The mutation can lead to a different phenotype of the patients. So this is why Rett syndrome patients are very, very different one from another. In addition, there is also the X chromosome inactivation, which lead to the mosaic pattern of cells and all the other things that I already explained.
Right. So we have another question. Sorry, if I don't pronounce properly your name. Is from Red or Death, and this is for Valeria again. Mm-hmm. And the question is: we found low NRF2 expression in autism brain, which parallels your Red findings. Dr. Richard Death is the person who made the question.
Thank you for the.
Do you agree with
comment.
it? Do you think there is a parallel situation?
In my findings, I couldn't show on the fibroblast. Actually, are quite different because I saw an increased translocation of NRF2 into the fibroblast, but without any a proper response upon pro-oxidative or pro-inflammatory stimuli. However, the other papers that I showed on rat mouse hippocampus showed an unchanged levels of NRF2 total and yes, in a basal condition. However, maybe we can write and talk together. And my colleagues, Alessandra Pecorelli and our PhD student are now currently investigating autism spectrum disorder. So maybe we can talk together by email.
Yes, thank you. In my opinion, autism is now a very promising target for NRF2 drugs, and actually some very interesting studies are going on, and preclinical data and also clinical data is going on. So the next question is again for Valeria. Valeria, you really made a great job today. This one is coming from Elisabetta Profumo, and the question is: which are the next steps to deepen your study the crosstalk between NF-kappa-B and NRF2 pathways?
Thank you for the question.
Particularly in the Red symptom.
Yes, thank you for the question. So now we are at the beginning of the study in the primary fibroblasts isolated from Red patients and control subjects. So we are going to do other experiments in vitro. In particular, maybe we are going to silence or inhibit NF-κB and see whether NRF2 can respond better in red cells. Otherwise, we can also switch our model for sure in vivo and see which are the responses at the microglial level and at the neuroinflammation level. We just obtained the approval to study in vivo, so we are very, very happy to do it.
So just in case that I can help on this question, with Isabel Lastres here, we reported that LPS induces two waves of different signaling pathways. In the first wave, you get a lot of inflammation and procytokines, increasing in the level of pro-inflammatory cytokines and these kind of things, but also NRF2. Yes. And in the second wave is when you get the feedback mechanism related to NRF2. So the problem is that in your model and in any other neurodegenerative condition, you have everything mixed. So most likely you will have both dysregulation of the NF-kappa-B system and also the NRF2 system.
Yes, I noticed this in my results. Yeah, thank you.
Okay, so we have Giovanni. Giovanni, thank you very much for joining us today. The question is really interesting program, and my apology, but needed to leave. Okay, Giovanni, no problem. Thank you very much. Another question from Richard Death. Dimethyl fumarate inhibits the NLRP3 inflammasome as well as the NRF2. How can you distinguish these two actions? I think this is a very important question, but mechanistically it is easily to answer, but from the point of view of pharmacology, target pharmacology is complicated. Is anybody interested in giving an answer?
Maybe I can just give a short answer in this because I'm also teaching about inflammasome and because I know about NF-kappa B and NRF2. The thing is, for the activation of inflammasome, first you have to activate NF-kappa B system and then you have a second hit. And in this case also, if you have that NRF2 inhibits NF-kappa B, you have an inhibition of inflammasome. That's maybe the crosstalk between NF-kappa B and NRF2. The thing is, it's not easy to distinguish between both actions because they are together really involved. So it's some kind of difficult to just make an analysis of inflammasome and NRF2. Maybe, as Antonio said, you have two waves, and in a very short time, you have an activation of NF-kappa B, and in a second time, you have an activation on NRF2. That's maybe a way to kind of analysis this action. But as I mentioned, there is a really, really tight crosstalk between both pathways, and it's not so easy to distinguish the effect of dimethyl fumarate on that.
So what we found is that in the NRF2 knockout animal, still there are NRF2 independent effects which which attenuate inflammation. And this is actually work that we did together with Isabel several years ago. So for sure there are NRF2 independent effects of dimethyl fumarate on inflammation. But how to discriminate this from the from the clinical point of view, it will be complicated. All right, next question also from Richard. ATP activation of inflammasome causes a dramatic decrease in GSH. And there is a paper here. So is there any specific question?
I think he's giving information about papers related to that.
Yeah, I meant that to be a guide to other people who are interested in the glutathione, because there's a 90% decrease in GSH upon activation of the P2X7 receptor. It's a very dramatic decrease in GSH, so obviously, and NRF2 would have the counter effect.
Yes, very good point. The only thing is that the pool of GSH is very high, probably roughly 1 millimolar. So it is also very sensitive, because of course it is not easy to deplete this pool unless you do something like hydrogen peroxide, something really very, very harmful. So small changes in the pool of GSH will certainly have a great impact on glutathione of proteins, including these ones, for sure.
I recommend that paper because it's an unbelievable decrease in GSH, indicating that it's a trigger. And I think the general point here is that the NLRP3 inflammasome is, in fact, a sensor of oxidative stress. That is its role. And as such, you can understand the glutathione, NRF2 would be an important regulator of it.
All right, we will read the paper. And maybe would you like to comment on the next paper that you mentioned related to autism?
Again, that was a follow-up to Dr. Cordon, because the Rett syndrome and autism are parallel. It's a autism postmortem brain levels of NRF2 are low, remarkably low, and neuroinflammation is probably facilitated by the low NRF2 activity, which seems again to parallel, although different original mechanisms, but seems to parallel the Rett finding that she so nicely presented.
Thank you so much for the paper. I will for sure study it. Thank you.
Okay, I don't know. I don't see any other questions in the chat. So if there are not more questions, I would like to pass the word to Brigitta, just to again thank her for this effort to organize these webinars. Brigitta, which is next?
Oh, the next, we have to decide. The next week, we will have to decide about the topics and the speakers. It will be a surprise for our attendance.
Okay.
So thank you so much, the speaker, for the interesting talks and the clear answers. Thank you everyone for participating to this webinar. I hope this webinar has been informative and helpful. And also I want to remind that recorded webinars are always available on our website and on the cost section website, BenBetFar. So share this information with colleagues and students. And I'm looking forward to keeping in touch for the next events.
Okay, thank you very much and bye-bye.
Thank you. Bye.
Thank you. Bye.
Bye bye. Thank you. Bye bye.